Hello, welcome to our biology video. Hope you enjoy our simple illustration and hope we add something new to your information. So, what will we talk about in this video? Our topic will be the DNA. DNA is the hereditary material that composes genes which encodes the traits in organisms. In this video, we will talk about its origin, structure, and its replication. Like for example, your hazel eyes, your sister's freckles, and your friend's Monica's stunning curly hair are all encoded in their DNA and genes. DNA encodes for all different traits, which causes everyone to have a unique shape. The discovery of DNA as genetic material has fueled molecular genetics research to this day. The work of several researchers has led to our current understanding of how DNA functions as the means of heredity. The researchers used varying types of evidence to build our knowledge of the function of DNA, and here's the timeline DNA discovery, which started in 1928 with Frederick Griffith, who discovered the transforming factor. The discovery of DNA as genetic material started in 1928 with the work of Frederick Griffith, who hypothesized the presence of a mysterious substance that could transfer genetic material from one cell to another. He called this unidentified substance a transforming factor. And how did he find out? Here's the story. Griffith performed an experiment. Griffith used mice to examine two types of strains of bacteria. The S strain was deadly and was called S because it had a smooth outer surface made of protein, and the R strain was harmless and called R because it had rough outer surface. He injected the mice with bacteria, and of course the S strain killed the mice, while the R strain kept the mice healthy. Griffith wanted to know what part of the S strain was killing the mice he used. So he heated the bacteria to remove the protein shells, and then he injected the mice. And yes, the heated strain didn't kill the mice. He next combined the R strain with the heated S strain and injected the mice. And surprisingly, the mice died. That's weird. Examined the dead mice, he found living S strain in their bodies. So he thought and thought and thought, and he found it. This meant there was a substance to heat treated S strain transformed to the living R strain that eventually caused the mice to die. He called it transforming factor. After Jeffrey's discovery came another scientist. In 1944, Oswald Avery, who discovered that transforming factor is DNA. Oswald Avery, Colin MacLeod, and Macklin McCarthy. Oswald wanted to discover the transforming factor is DNA, RNA, or protein. So they made an experiment. In different groups of heat-killed virulent S cells, only DNA, only RNA, or only proteins were systematically destroyed. Only the group in which DNA was destroyed failed to transform non-virulent bacteria into virulent bacteria. Therefore, it was clear, now DNA is our man. However, many scientists did not accept these results, and other teams of scientists continued to perform experiments. It's not until 8 years after Avery's experiment, in 1952, that the controversy was settled by Alfred Hershey and Martha Chase. Hershey and Chase ran two experiments using viruses. In the first experiment, they labeled protein shells in the virus with sulfur atoms. In the second experiment, the DNA was labeled with phosphorus atoms. When placed with bacteria, the virus infected their DNA into the bacteria. Then all the material except what was inside the bacteria was removed. Hershey and Chase looked to see where the labels were found. The sulfur labels on the protein were found outside the bacteria, only the phosphorus was inside. And what do they get from this? This proved that it was DNA, not proteins, that was transmitted to the bacteria. The work of Griffith, Avery, Hershey, and Chase provided basis for today's understanding of the structure and function of DNA. DNA is composed of two strands of nucleotides and that hydrogen bonded to each other, forming double helix. James Watson and Franz Crick could find out the inner structure of the helix. In DNA, nitrogenous bases can be double ring or single ring. Double ring structure called purine always form bonds with single ring structure called pyrimidines. Notice that the purine adenine A pairs with the pyrimidine thymine T, and the purine guanine G pairs with the pyrimidine cytosine C. Complementary base pairing is only one of the consistent properties of the DNA molecule. Symmetry is also another property that is consistent through DNA. Look at the shoes on your feet. Each shoe has a toe end and a heel end that are very different. DNA has a similar directional structure, that is, the two ends of a singular standard differ, like the ends of your shoes, and are antiparallel. Antiparallel strands are arranged like the pair of shoes. A 3 end if one strand is linked to a 5 end if the other strand. 3 means 3' hydroxide group and 5 means 5' phosphate group.
Parent strands of DNA are antiparallel. They are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments.
Now, how did the DNA replicate? And we mean by replication when a molecule reproduces itself giving out two identical molecules. Oh, Melson and Stahl found out. By tracking the amount of radioactively labeled nitrogen-15 DNA, Melson and Stahl were able to determine the precise mechanism of DNA replication. In their experiment, each strand of DNA molecule was radioactively labeled before the replication process. Since the DNA molecule was capable of base pairing, each strand should be capable of being a template for the synthesis of a new strand. The final two helix each contained one original labeled strand, one new strand, which makes DNA replication a semi-conservative. Now let's talk about DNA replication steps. Replication has two main steps: the assembly of the double helix done by helicase enzyme and addition of free nucleotides done by DNA polymerase. Researchers discovered the helicase are enzymes that separate double-stranded DNA into two strands of nucleotides. Each of these template strands will produce a new DNA molecule identical to the original by the addition of free nucleotides from the environment.
Replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain.
DNA polymerase is one of the enzymes that catalyze the addition of complementary nucleotides to the template strand of DNA at the replication forks. The addition of nucleotides to the new single strand, known as extension, always progresses from its 5' end to its 3' end. The extension of the 5 to 3 strand of DNA, called the leading strand, is continuous. In the other strand, called the lagging strand, extension to the 3' direction in backwards, or discontinuous because of its orientation. The lagging strand must add nucleotides at the replication fork. But how? The answer is that the lagging strand builds short segments of DNA in the 3 to 5 direction, but works backwards, installing the segment in the 5 to 3 direction. These result segments complemented DNA in the lagging strand are called Okazaki fragments. Each Okazaki fragment requires its own primer. As a result, the segments of DNA must be linked after they are installed. DNA ligase catalyzes the bonds between Okazaki fragments on the lagging strands. And here's a simple illustration. DNA replication occurs at relatively rapid rates in eukaryotes as compared to prokaryotes. Prokaryotes generally have a single replication bubble on their single circular chromosome. This replication bubble has two replication forks. By contrast, the chromosome of eukaryotes have a linear structure so that multiple replication bubbles, each with the two replication forks, open on a single chromosome, speeding up replication. When the two strands of the original DNA unwind and unopen, the unopened section of the DNA must twist. Topiose pairs are enzymes that clip the DNA, allow it to untwist, and the fuse of the DNA back into the place. During the process of DNA replication, errors can occur. If the error is correct, there is no obvious phenotypic defect. If the error is uncorrected, like the one that causes albino coloration, this is a mutation. A mutation such as the one that causes animals or plants to be albino. Fortunately, there are ways for DNA to correct errors, including proofreading and mismatch repair. DNA polymerase proofreads the complementary base to scan for errors. If an error is found, the incorrect base is removed and a new base is inserted. And now, ladies and gentlemen, our video comes to an end. Hope it was blissful and enjoyable for you as it was for us. Thank you for watching.